This episode is brought to you by forestbars.com, natural supplements for human enhancement. Forest produces full spectrum, bioactive, mushroom infused chocolate with powerful benefits, sustainably sourced and served. Receive 10% off your first order with code 0hour10. Good afternoon, everybody, and welcome to the webinar. It is my honor and a privilege to introduce our speaker today, Dr. Dolen. Dr. Dolen earned her MD Ph.D. at Brown University and the Massachusetts Institute of Technology, then completed postdoctoral training in the Department of Psychiatry at Stanford University, where she did paradigm-shifting work on the neural circuits underlying social reward learning. In 2014, Dr. Dolen began her faculty position in the Department of Neuroscience at the Johns Hopkins University School of Medicine. Dr. Dolen's work has been featured in Nature, the world's leading multidisciplinary science journal. The Dolen Laboratory has made the groundbreaking discovery that MDMA can reopen a novel critical period of brain plasticity in mice. Building on that discovery, Dr. Dolen has initiated the Fathom Project. I would like to thank my co-host today, Adam from Psychedelics Anonymous. And with that, we will get started. Dr. Dolen, welcome. And can you tell us a little bit about your current research?
Okay, thank you for having me. It's a pleasure. Yeah, so my lab, we started out being mostly interested in social behaviors and diseases of the social brain. And in the process of that, we discovered that, like many other things in the brain, social reward learning is something that is regulated across development. So you're much better at being able to learn from your social environment when you're young compared to when you're old. And that phenomenon of, changing your ability to learn across development is something that neuroscientists call critical periods. So there are lots of critical periods. There's a critical period for language learning that most people are pretty familiar with, but also critical periods for things like organizing visual information processing and somatosensory information processing and motor information processing. And my lab discovered a new one, this critical period for social reward learning. And then we kind of, got lucky, I'll be honest, when we discovered that the psychedelic drug MDMA is able to reopen this critical period. And basically what that means is that, if the adage, you can't teach an old mouse new tricks is true, then when we gave them MDMA, we were able to teach old mice new tricks. And that was super exciting. But at the time, we thought that was really about the fact that MDMA is this incredibly pro-social psychedelic. So it's different from other psychedelics in that it's been characterized as an empathogen, or some people call it an entactogen. And really, this reflects the difference between MDMA and other psychedelics in that it has this very pronounced pro-social property, which we've also looked at in octopuses, and, it's conserved across evolution. It's really, a feature of MDMA. But other psychedelic drugs like LSD and psilocybin, while they may make you feel more interconnected with the universe, nobody's doing a 60-person cuddle puddle on LSD. And so we had this intuition that the reason that MDMA was working so well to reopen our critical period was because of that pro-social quality. And what we've discovered more recently is that that's just not true, and that in fact all of the psychedelics are able to reopen our critical period. And what that suggests to us is that the ability of psychedelics to open critical periods is not specific to only ones that have this pro-social property, but it might be a general property of psychedelics. And that's gotten us really, really excited about what that means for not only how we understand what psychedelics are doing in the brain, but also in terms of how we might use them in a clinical therapeutic setting, how we might branch out from what we're doing right now to use it in other clinical settings and also non-clinical settings.
Right. Could you go a little bit deeper into the critical periods in a social context and how that works?
Yeah, so I think just to back up a little bit, maybe it gives some idea about why we thought there might be a critical period for social reward learning to begin with. So I think that, it's probably, if anybody, has awful memories of what it's like to be a teenager like I do, it's hard. It's something that takes a lot of energy to constantly care about what people think about you and to try and fit in with your social group and define yourself based on those kinds of feedback that you get from other people, whether you're, in my case, whether your acid wash jeans are the exact right shade or whatever it is, right? And so that's a really emotionally draining time, but it's an important time where you learn what the rules of engagement are for your particular social group, right? So if you're a nerdy scientist, you learn that, it's okay to carry around your graphic calculator, but, if you're trying to be one of the cool kids, that's probably not a good idea. And so I think intuitively we had that sense based on the human literature that, humans undergo these radical shifts in how much they care about their social environment between when they're kids and when they're adults. And in fact, I think as we get older, we kind of feel relieved a little bit that we don't have to care so much about social interactions. And we still like social interactions, but, after a week at a conference or something, most of us as adults favor that quality alone time where we get to recover our emotional resources. And so what we think is that the reason that critical period closes is because it's just takes a lot of energy to learn that much from your social environment. And so once you've learned the rules and you're living in a stable social environment, it makes sense to divert those emotional resources elsewhere. And that's why we think in general, critical periods close, and specifically this critical period closes.
It's fascinating. I have heard you speak before on this concept of the master key. I'm wondering if you could explain how psychedelics and these particular group of therapeutics can be applied with respect to historical use.
Yeah, so the concept of the master key for reopening critical periods is pretty old. I think basically since 1935, when Konrad Lorenz described the first critical period, people kind of had this intuition that if there was a way that we could reopen critical periods, that would be very useful for our ability to intervene in therapeutic contexts. And probably the best example of that is the ocular dominance critical period, where, visual information processing gets reorganized in early development, but as the brain matures, it's no longer able to do that. And the therapeutic consequence of that critical period is that if you're born with bilateral cataracts and then they aren't removed until after that critical period is closed, so if they're not removed before the age of 4 or 5, then even if you remove the cataracts, you will be blind forever because the brain is no longer able to rearrange its circuits and connection probabilities and strength of responses to that change in the environment, namely removal of the cataracts. And so, people have been on the hunt and, 3 Nobel Prizes have been given looking for that master key. And yet nobody's been able to find something that would be clinically useful. People have found manipulations that are, not something that we would ever really do in a clinic. And also people have had this sort of lingering worry that, anything that might be a master key would also potentially harm the brain, right? So you could imagine based on the mechanisms that people have implicated in the closure of critical periods, that a master key could, cause structural instability for the brain, or cause there to be amnesia, or cause there to be seizures, right? And so people have been, excited about the possibility of finding a master key, but also worried about what that might look like in a therapeutic context. So that's happening, in neuroscience where I live, kind of in parallel as this, psychedelics renaissance, if you will, is happening in the clinical setting where, psychedelics have been used for millennia in religious and medicinal practices outside of the sort of Western culture. But, more recently, they've been showing these remarkably promising therapeutic results in clinical trials for diseases that, on the surface don't really look very much like each other. You've got PTSD on one hand, and depression, addiction. They seem like they're all very different, and yet psychedelics have been showing promise for treating all of those diseases. And yet we really don't have any understanding of how that's working. People have tried to focus on receptors, or taking sort of the traditional receptor pharmacology approach to try and untangle what it is that these drugs are doing in the brain, to try and explain their mechanisms. But the explanations that people have come up with so far using that kind of traditional approach have failed to recapitulate some really important properties of psychedelics therapeutic action, which I can kind of outline in more detail. So what we think we've discovered is that, this ability of the psychedelics to reopen our critical period, if it extends to all the other critical periods, and it turns out that psychedelics are that master key that we've been looking for, it not only, solves this long-standing problem in neuroscience, but we think explains the diversity of diseases and some of the other clinical features of how psychedelics seem to be working in a therapeutic setting. And so we're really excited that this discovery might have sort of bridged that gap between, the excitement over psychedelics as therapeutics and the hunt for the master key.
Right. You mentioned before that you work with MDMA. Why did you choose that? Or why are you working with that over other psychedelics?
Yeah, so we started with MDMA because of all the psychedelics, it's the one that has the acute subjective effects that we can measure in a mouse, right? So, MDMA, just like in humans, in mice, causes, an acute prosocial behavior that we can measure, right? Whereas we don't really know necessarily what a mouse hallucinating looks like. It's a little bit unclear whether or not the distribution of serotonin 2A receptors in the visual cortex of a mouse makes that an easy thing to answer. And so we started with MDMA purely for the practical reason that, unlike dissociative properties of some psychedelics, or hallucinogenic properties of others, or onirogenic properties of others, this empathogenic property is something that we can measure in the lab. And that's really why we started with that one. We did for a minute think that maybe we could use octopuses to look at hallucinations, because, we thought, well, if an octopus is hallucinating, then, and they are able to do these color changes, camouflage skin changes, that whatever they're hallucinating would show up on their skin patterns. But the first attempts at those experiments, not by us, but by other people, kind of suggest that it's not going to be that straightforward.
And with that, I think it would be great if you could speak a little bit more about the octopus study, and specifically how that ties back into the default mode network. And for those listening, it would be helpful to explain what the default mode network is.
I think some of the earliest human research since the Renaissance of psychedelics has really basically looked at giving people psychedelics, LSD or psilocybin, and then taken brain images of people's, brain activity in response to those psychedelic drugs. And the reason so fMRI imaging, what it's looking at is it's measuring blood flow to different brain regions. So it's not really measuring neuronal activity per se, it's measuring how much energy active neurons need by requiring more blood flow to that area. And what was discovered is that a series of brain regions that are interconnected to each other seem to be, basically their activity seems to be being reduced in response to psychedelics. And those brain regions that are connected in that way are called the default mode network. They include the dorsal medial prefrontal cortex, a number of other brain regions. And the reason they're called the default brain network is that it was noticed years ago that if you tell somebody in an fMRI machine to do, let's say, a math task, right? Like an addition task. And then you need to take baseline measurements between tasks in order to sort of calibrate the response to what a person is doing, what the brain activity is doing when they're not following the instructions about the math problem. Then you just tell them, don't think about anything. And when they are not thinking about anything, in quotes, a set of default brain regions lights up, and those default brain regions that light up have been called the default mode network. Now, there are a lot of explanations for what the default mode network is. Some people think it's, important for social information processing. Matthew Lieberman has, a beautiful set of studies and a book about this. But, Robin Carhart Harris suggested that maybe this is important brain regions for ego, and that when you see that the activity of those neurons go down, that is a reflection of the ego dissolution property that some people report on psychedelics. So this is a kind of explanation that, I feel like, the data is really hard to interpret because fMRI imaging by its very nature, doesn't have the spatial and temporal resolution to be able to minute by minute or second by second or millisecond by millisecond, which is, where we're at with recording from neurons, to really understand what psychedelics are doing to the brain. But nevertheless, I think we're all in the habit, whatever technology you're using to measure brain activity, we are all in the habit of identifying a brain region either by imaging or in my case by molecular genetic manipulations and saying this brain region, if we silence it, if we activate it, if we manipulate the receptors in it, is responsible for activity of psychedelics X, right? And that habit that we're in is because we're trying to explain the phenomenon in terms of brain anatomy. And so what was so, I think, surprising and unnerving about the octopus experiments is here is an animal that does not have a default mode network. It doesn't even have a cortex. It doesn't have a nucleus accumbens. It doesn't have an amygdala. Its brain is organized in a totally different way that's much more similar to a slug than it is to a human or a mouse, right? And so despite this difference in brain anatomy, what it does have at the molecular level is a binding pocket for MDMA, which is the binding pocket for serotonin, which turns out to be a really old neurotransmitter. And it turns out that just by having a similar enough binding pocket for serotonin, MDMA is able to induce prosocial behavior in this animal. And so it tells us a couple of things. One, it tells us that even though serotonin's been implicated in almost every behavior you can think of, the function of serotonin to regulate sociality is very old. So, the last time that humans and octopuses shared a common ancestor was about 650 million years ago. And just to kind of put that into context, we're more closely related to a starfish than we are to an octopus. Like we're closer to dinosaurs than to octopuses. Okay. And so they're really different from us. And yet the mechanism seems to come down to the level of molecules. And it suggests that all of this obsession with anatomy and circuit mapping and, what I call blobology, for the fMRI imaging stuff, is maybe a red herring, and we're not really understanding as much about how these things work as we originally supposed.
Great. So I'm aware of your background research with autism. Do you expand on how that relates to the current work that you're doing?
when I was a PhD student, my PhD advisor and I had what we thought was a great idea for what causes autism. It was called the mGlu-R theory of Fragile X and autism. And basically the idea is that we were studying the genetic causes of autism and at the time there were only 7 known genetic causes. Now there's close to 1,000. And so, but, the one we were studying was, the main first identified, still the most common identified cause, Fragile X. And what we had the idea is that in Fragile X, when there's a mutation in that FMR1 gene, it causes a biochemical imbalance with a receptor called the mGlu-R5. And that imbalance is sort of responsible for all of the disease symptoms that people have. And that if we could just restore that balance or fix that imbalance, then we could cure the disease. And so we did those experiments in mice. Something like 26 other labs reproduced our findings in mice, and everybody was super excited. And we went into clinical trials, and Novartis and Roche and Pfizer and Seaside Therapeutics, a bunch of companies invested hundreds of millions of dollars to run these human clinical trials, and then they failed. And this was like a big disappointment in the field. And I think a lot of the clinicians who were running these trials kind of went, well, you see, we told you so, mice aren't humans, and that's why these failed. And we just weren't, my lab, we weren't satisfied with that explanation, because there were a lot of other differences between the human clinical trials and the preclinical studies done in mice. And the one that really stuck out for me is the fact that all of the studies done in mice, we did this intervention with the mGlu-R5 very early in development, whereas the human clinical trials, the intervention was done in adults. And that was by design because, ethically, you have to start with adult patients. And basically, we sort of have this hypothesis that the reason that the human clinical trials failed is because by the time we did the intervention with the mGlu-R modulator, the critical period, the relevant critical period, had already closed. And so what I'm imagining is that just like that blindness situation, in some sense, you could think of what's happened is that the mGlu-R5 negative allosteric modulator is like removing the cataracts. It can work if you do it early enough, but if you wait until the social critical period, in this case, is closed, then fixing the imbalance doesn't fix the behavioral problems that define autism. So what my lab is working on right now is testing the idea that if we combine psychedelics with the manipulations to restore this biochemical imbalance, then we can return the brain to the normal functioning and reopen the social critical period to give people the opportunity to learn from their social environment with the biochemical imbalance sort of restored.
Perfect.
Wow. Now, with your current lab, the Fathom project, I understand that is an acronym. Can you tell us what Fathom stands for and how that ties into the importance of setting, and the pairing of something with psychedelic therapeutics?
Yes, great. So Fathom stands for Psychedelic Healing Adjunct Therapy Harnessing Open Malleability. That's I know it's a mouthful, but just so you know, people who run clinical trials love to have cool acronyms. The Fathom trial we felt like it, really rolls off the tongue, even though the acronym is based on, a wordy jumble of things. But the reason that we focused on Fathom or this psychedelic healing adjunct therapy is really because we think that based on our results in mice, that reopening the critical period for social reward learning is context dependent, and that this context dependence of psychedelics' ability to reopen the critical period captures an important clinical element of how psychedelics seem to be working, namely that the therapeutic effects are also context dependent. So, you can't go to a rave and take MDMA and expect that your PTSD is going to be cured. The PTSD clinical trials really emphasize the importance of being in the right mindset or setting, in this case, psychotherapy for the PTSD. And they really emphasize the importance of that pairing of the psychedelic with the right set and setting, or as neuroscientists, we call that context, right? And so the context dependence of therapeutics, effects of psychedelics, nobody else, to my knowledge, has been able to recapitulate that property in an animal model. So we're really excited that we can recapitulate this. And what this suggests is that the reason that, for example, clinical trials where there wasn't particular attention paid to the set and setting, right? So in the clinical trial where they looked at the effect of psilocybin for depression, that study, there were psychotherapy, it was available, but it was really more focused on the idea that the psychotherapist was there in case somebody had a bad trip, but really psilocybin was being used sort of like a next generation antidepressant, works faster. And the results were sort of underwhelming, right? Like the amount of improvement was basically the same as SSRIs for depression, right? So if you compare that trial to the remarkably efficacious and better than psychotherapy by itself, better than SSRIs by itself, results from the MDMA-assisted psychotherapy trial for PTSD, I think that gives us a clue that the context or the set and setting is so important for the therapeutic effects of these drugs. And so we think that the adjunct therapy element of the Fathom trial is really, really important. So now if we're right that this context dependence of social critical period opening, namely that we can only open the social critical period if we give the MDMA in a social context, but not in an isolation context, we think that social versus isolation is important because the critical period we're trying to open is a social critical period. But as we start thinking about what other critical periods we might be able to open, we are most excited about the possibility of opening a critical period for motor learning after a stroke. And in that case, our hypothesis is that the social context won't matter, but the motor context will matter. And so we're sort of imagining that the adjunct that psychedelics will be paired to in that situation will be more like physical therapy rather than psychotherapy.
Cool. So where do you see your work moving towards in the future? Like, where are you taking your research and the outcomes that you're achieving?
Basically, there are three major areas that we're going in. So one is to try and reopen as many critical periods as we can. So we have collaborations with labs like Steve Zeiler's lab and John Krakauer's lab that, focus on the motor critical period. We have another collaboration with Paul Worley and Alfredo Kirkwood, who are looking at somatosensory and visual critical periods. And we have another collaboration with Jeremiah Cohen, who's looking, when we're trying to define some cognitive critical periods. So basically, if you're a listener out there and you happen to be a neuroscientist who has a critical period that you're dying to reopen, please reach out. We are on the hunt to try and open as many as we can. I mean, I'm willing to be really open-minded about this. I also think we could probably even open a critical period for allergy, and I think this will have huge implications for a whole variety of therapeutics. The other major area that we are focused on is that even though this critical period explanation we think offers a lot, we still feel like there are a couple of pieces that are missing. And so we are also doing a deep dive on the mechanism and trying to understand at the cellular level, how does the neuron know that it's in the right context, right? Like, what is the cellular mechanism for that context? And I won't bore your listeners with some of the theories that we have on that, but some of the psychedelics, we have easy answers, right? Like the NMDA receptor, which ketamine binds to, is a coincidence detector. Naturally, that's what it does. And so we like that that seems like a easy-ish explanation, but the other receptors don't do that. So we're looking for what are the cellular coincidence detectors that could potentially explain that context dependence, because I think that will go a long way to answer the problem of how it is that psychedelics are able to circumvent this melty brain problem that I mentioned is, plaguing or worrying us about finding a master key for psychedelics. And then the third major area that my lab is focused on is, and this is, I always joke that my lab is, basically the psychedelics are the really conservative project in my lab, and the out there project is the octopus. And we are really excited about the octopus because last year we published a paper in collaboration with Woods Hole, closing the life cycle of an octopus. We're getting very close to finishing up the genome. And then when we can do that, then I really feel like the octopus is going to be the next frontier in neuroscience, because I think it can offer us all kinds of opportunities to study an alien intelligence right here on Earth and start to get after the sort of hard problems of neuroscience, like what is consciousness? And if we can do that with psychedelics in a species where we're not going to be distracted by sort of brain requirements that are sort of evolutionary accidents rather than true generalizable motifs, then, we're off to the races. So, yeah.
So what would you say are potential impediments or pitfalls and setbacks to the progress that you're making as you push forward with these things?
I think that one of the things that has me the most concerned is that as psychedelics have gotten so much more attention, and so many drug companies and so many investors see a potential for making a lot of money off of this psychedelic revolution, I think the intuition has been to use the same kind of playbook that big pharma and drug development has used for the last 50 years. Namely, we want to, we've given up on the idea that we could ever cure depression or PTSD. We just want to treat the symptoms, and we're okay with making people have to take a medicine for the rest of their life. In fact, it's a pretty good business model, right? If you medicalize your patients. And so I think what's happened is that with the enthusiasm around psychedelics, there's been this rush to try and figure out the simplest mechanism that we can model in a dish, and then develop as many different psychedelics that mimic that feature, whatever we've decided is the equivalent of curing PTSD in a dish, right? And then develop a whole bunch of new compounds that we can patent, and not really, and especially focus on things where we can get rid of the psychedelic side effects, and we can kind of focus in on creating a medicine that doesn't require all the support and all of the therapy associated with, these psychedelics, which many of them are very long acting, right? And so some people call that effort, that push, the trying to democratize psychedelics. And, I'm really worried that it's not really democratizing psychedelics. It's A, monetizing them, and B, potentially turning these drugs that have been very effective, at least in sort of anecdotal reports and, a handful of small trials, for curing drug addiction. For example, ibogaine has been remarkable for curing heroin addiction so far, and then essentially engineering them back based on ideas like psychoplastogen, a word that I hate and I'm like really hardcore want to push against, and turning them into drugs like cocaine or methamphetamine, which are hyperplastic but addictive rather than curing addiction. So I think that's like one major pitfall is that if we treat these drugs like they're just another, next generation SSRI, next generation diazepam, next generation whatever, then we're going to miss not only how they're working, but we have the potential to do great damage to people who are seeking out these drugs for treating addiction, depression, PTSD, et cetera.
Couldn't agree more.
we see a rapidly shifting landscape, especially with the recent decriminalization of psychedelics in the state of Colorado. My next question pertains to safety. For those that are seeking psychedelic experiences, physicians are not able to prescribe psilocybin as there's not an FDA approval. And the current avenue that is accepted from a medical legal framework is through clinical trials. However, there are those that don't have access, maybe constrained by geographics, and those that are seeking ceremonial experiences outside of a lab, perhaps in Costa Rica, Peru, or even underground. So with that, what advice or words of caution would you lend?
When we're thinking about psychedelics, we need to recognize that even though these have been used in traditional settings for millennia, as I mentioned already, the context that they were being used in is very different from the context that we're using them. And they are very powerful drugs. And we need to have respect for the incredible power of these drugs and not use them flippantly. And so my caution comes from some data that we have that shows that if reopening critical periods with psychedelics is not just something that happens and then they close right back up. Indeed, we have evidence that how long the critical period stays open after the psychedelic is sort of directly proportional to the duration of the acute subjective effects, right? And that proportionality means that something like MDMA, for example, which its acute subjective effects last for about 5 to 6 hours in humans and in mice. And in mice, at least, it keeps the critical period open for 2 weeks after the acute subjective effects have subsided. And so what that means for humans is that MDMA is probably keeping this critical period open for at least 2 weeks, but it could be as long as 2 months, right? It's hard to translate mouse time and human time, especially when we're talking about developmental processes. And so if you think about what it means to restore the brain to this state of vulnerability and sensitivity to the world that makes you sort of childlike in your vulnerability, right? We should all be concerned that we are restoring this childlike state to people and then just kind of not safeguarding them the way that we would safeguard a child who's still in the open state of their critical period for social, and that we need to be building in safeguards for people. And if they're using them recreationally, to kind of keep in mind that these are powerful drugs and you probably wouldn't want to be doing them and exposing yourself to environments that you wouldn't expose a 5-year-old to, right? And being aware of the fact that you are going to be vulnerable and sensitive in that way for an extended period of time. So, and I think that, just as a cautionary, I think this kind of explanation also explains why, for example, Charles Manson, who gave people who were in his cult psychedelics, every day, and then indoctrinated them into believing that he was their, religious leader, and then convinced them to, go out and murder people. This is exactly the kind of thing that we would predict based on doing psychedelics in a bad context, right? I mean, doing psychedelics with a psychopath, I would say, counts as a bad context. And the sort of potential consequences of that kind of unthoughtful use, or, not understanding what they're doing and using them as if they're just totally benign and harmless. And it's not that they're necessarily harmful. I also am not convinced that they are necessarily good. They are sort of neutral, and the valence of their properties has everything to do with the context that they're being used in.
Very, very important note there. Thank you for that. A lot of the current work and literature to date focuses on mental health. You mentioned how ibogaine has a lot of efficacy for opioid addiction and withdrawal. The Veterans Affairs right now is looking at MDMA for PTSD. I'm wondering if you can comment on indications beyond the mental health space, specifically neurodegenerative disease, and how psychedelics can play a role there.
I'm not really sure what the opportunity for neurodegenerative disease is. I know that some people are excited about that. My intuition is that for neurodegenerative disease, we're looking at potentially a different mechanism, right? So I think that with neurodegenerative diseases like Alzheimer's and Parkinson's, if there is a significant element or component of inflammatory response or, repair responses, there are some studies that suggest that some psychedelics, but not all psychedelics, have this anti-inflammatory property. And this is the work of Charles Nichols. It's really not what we're focused on. I think the same is possibly true for headaches and migraine. I feel like those might be a different type of property of psychedelics that's being used for that. On the other hand, I think that there are a number of diseases. I already mentioned stroke, but traumatic brain injury, language learning after cochlear implant, blindness, I already mentioned, autism, I already mentioned, where there is a relevant critical period that we would want to reopen. I also think, we could potentially go in the direction of, opening up language schools and also trying to branch out into understanding how other practices have shared properties with psychedelics, like the mystical experiences that people report and consciousness expansion. Those are other possible areas, but neurodegeneration per se, it's not clear to me anyway at this point. I mean, maybe someday in the future it'll become clearer, but right now I don't have a intuition that they will be particularly useful for diseases like Alzheimer's or Parkinson's, at least using this mechanism that we have identified. There may be other mechanisms.
And a follow-up to that, do you think that there is a role for disorders of consciousness, such as post-cardiac arrest, anoxic brain injury, or diffuse axonal injury, or persistent vegetative state? Because the current standard of care doesn't seem to get to any improvement in those conditions.
Yeah, I mean, that's going to be tricky. We definitely have looked, in mice anyway, at whether or not anesthetic doses of psychedelics like ketamine are able to reopen this critical period, and they don't. And we think that the reason for that, it could be one of two things. One is that the dose response of the brain to ketamine is complicated, so more isn't always better. And it could be that at higher doses, the ketamine is able to bind to other receptors that are sort of counterbalancing whatever the psychedelic effects the lower doses are having. But what I actually think is happening there is that when an animal is anesthetized, they are not having a social context, right? The social context is absent when an animal is not not consciously aware of its environment, right? And so, or its social group. But, that doesn't mean that there couldn't be an opportunity, right? So we haven't done this experiment, but one sort of interesting thought experiment would be, okay, well, we know that social touch vibrates at a certain frequency. What would happen if we gave an anesthetic dose and then gave the animals social touch, we applied it to them in a way that's sort of artificial? If we could do that, then it would suggest, well, that maybe, in someone like chronic coma or, vegetative state, if you could kind of pair the psychedelic with some sort of stimulus to trigger the right brain circuitry for, that learned experience, then maybe, that's not going to be my first area to jump into, but it certainly, again, depends on the context. And the problem with coma and diseases of consciousness is finding a way of stimulating the right context or the right memory engram or the right circuit of neurons to get activated appropriately to trigger that learned response.
You mentioned before that not all neuroplasticity is good. Talk a little bit about how neuroprotection differs from that, and maybe with reference to the well-documented cocaine control.
Plasticity is kind of it's a word that's used in a lot of different ways by a lot of different people. As a synaptic physiologist who does whole cell patch clamp electrophysiology, what I mean by that is electrophysiological plasticity, although there's plenty of evidence to suggest that what happens to the electrical response properties at the synaptic level translates into changes in dendritic spine morphology on those little nubbies that you see on a neuron. And so that's called morphological plasticity. And then there's sort of, more general ways that people talk about plasticity, but I'm not going to focus on that, because the evidence that you're referring to about cocaine is that cocaine, like all drugs of abuse that have been studied, including methamphetamine, amphetamine, heroin, alcohol, nicotine, all of these drugs induce robust morphological plasticity, right? So you just see the neurons sprouting like crazy. And that property is shared by neurons in the nucleus accumbens and the medial prefrontal cortex, which are both part of what's called the mesocorticolimbic reward circuit. That's sort of the sex, drugs, and rock and roll addiction pathway, right? And those drugs cause that robust plasticity, which I call hyperplasticity. And that's thought to be the mechanism by which they are addictive, right? They sort of override the normal rules constraining learning and memory. So, a good example, since we're coming up on Thanksgiving, is that normal rewarding behaviors are context dependent. Our associative learning rules we really, really love Thanksgiving dinner, right? And turkey and mashed potatoes and pecan pie sounds delicious, right before you eat Thanksgiving dinner. But 30 minutes after Thanksgiving dinner, when you're stuffed, the last thing you want to think about is another piece of turkey, right? Because it just feels revolting to you, right? And another sort of way to think about that is when you learn, let's say you have a favorite sandwich shop and you're like, okay, when I'm hungry the next time and I'm in this neighborhood, I'm going to go to that hoagie shop because they make the best hoagie I've ever had, right? So again, it's context dependent, whereas drugs of abuse aren't like that, right? Like people who get addicted to cocaine or nicotine or heroin, they like cocaine in a house with a mouse, everywhere, right? It's not context dependent. It's overridden those rules of associative learning. And so I think it's really important to kind of keep that in the background when you hear people talking about the psychedelics might have this property of being psychoplastogenic, right? Like cocaine, heroin, they are great psychoactive plastogens, but that's not the good kind of plasticity. It's not the kind of plasticity that you want in a drug that's going to be used to treat addiction or depression or PTSD. And so what's more is that we have not been able to reproduce any of those findings that suggest that psychedelics might be hyperplastogenic. And we think the difference between the way we did the experiments and the way other people have done, the people who made those claims did the experiments, is that neurons in a dish, dissociated cultures of neurons, are essentially baby neurons living in a dish that doesn't have the appropriate sort of developmentally regulated matrix of proteins that we think are important for stabilizing critical periods. And so basically the rules that are constraining learning and memory to critical periods in an intact brain are absent when you try and model that in a dish. And so for that reason, we think that reports of psychedelics inducing hyperplasticity in a dish are an artifact of the preparation that they've used to measure those changes. And in vivo or ex vivo, the way that we do the experiments, there have been reports of plasticity. We have reported metaplasticity. Others have reported, calling changes in spine morphology, but they have really, the types of changes that they are reporting are more consistent with what we would call metaplasticity. In other words, very subtle changes that reflect a change in the ability of the brain to induce plasticity, not the plasticity itself. And so the way that we measure that is we look at oxytocin-induced plasticity. In adults, it's really hard for oxytocin. You can apply oxytocin. It doesn't induce plasticity in adults, but in juveniles, the same concentration of drug causes robust plasticity. And so what we have found is that psychedelics restore the brain's ability to respond to oxytocin, or rather, the metaplastically upregulate oxytocin-induced plasticity, right? And so that metaplasticity is very different and seems to obey all of the same rules of associative learning that makes it very different as a mechanistic explanation than, hyperplasticity, which we think is what drugs of abuse are doing.
Absolutely fascinating. Dr. Dolen, I want to extend gratitude for your time today, for this extremely enlightening conversation. Thank you to Voltura, Lewis Gale, Adam, and Psychedelics Anonymous for providing this platform. And of course, to Johns Hopkins for encouraging you to come join us today. With that, I'd like to give you the final word and shed some additional light with a take-home message for our listeners.
I don't know if I have a take-home message, but I would just say we're entering into a new phase of psychedelic research, which is an exciting time. And I think that what I have learned in my 30 plus years of doing neuroscience research and translational clinical studies, what I've learned from, doing work with trying to find cures for patients is that we really need to be careful not to get ahead of ourselves and make big promises that we can't keep, and also not learn from our previous mistakes, translating studies in mice to humans, and not get grandiose in the way that we just assume it'll all work out. I think we need to understand how these medicines are working in the clinical context that we are using them, and not be sort of overly optimistic about how we're going to be able to take the successes of thousands of years of use in indigenous communities or traditional settings, that transfer without understanding the mechanism is not going to be all that easy. And so I think we need to be excited. And, I think this is the new frontier, and I'm super hopeful that we're going to crack this and we're going to find the master key. But, I'm also trying to remind myself and my students and my postdocs and, patients that we are just at the beginning, and patience is required.
Very well said. Absolutely brilliant. For those of you who are interested and want to read more about Dr. Dolen's work, you can go to her website at dolenlab.org. Dr. Dolen, I look forward to seeing you in person at the MAPS conference next June in Denver. I wish you well, and thank you again so much for your time today. Thank you.
It's been a pleasure.
This episode is brought to you by forestbars.com, natural supplements for human enhancement. Forest produces full-spectrum, bioactive, mushroom-infused chocolate with powerful benefits, sustainably sourced and served. Receive 10% off your first order with code 0hour10.